And now it's my honor to introduce Tom Gajewski, a long-standing friend from University of Chicago, a good tennis player, and I would say that he loves Mediterranean as his wife is Spanish, so he loves good weather and he's happy to be here from cold Chicago. Please, Tom.
So guilty as charged. All of those things are true. So thanks so much for the invitation. The task that was given to me was the future of, I think, cancer immunotherapy. I'm going to start with a little bit of the historical past to put it in context. And what I want to try and illustrate is how going back and forth between patient material and mechanistic laboratory studies is enabling identification of new targets and new therapeutic approaches. So hopefully this feels fresh and a few new things, a little bit like Sergio's talk. I have to thank people for the hospitality. We had a bit of tourism as part of this trip, and I much appreciate that. And I'm wondering if after 2 days of a meeting, if you feel a little bit like these sleepy cats right now, but we'll see if I can stimulate some interest. I have some disclosures, and maybe we can launch in then, which is going back more than 15 years, we've been thinking about the biology of the tumor microenvironment and how it's the ultimate arbiter of what happens with the anti-tumor immune response. And the initial observation that a major fraction of patients already has at baseline what we had called a T-cell inflamed tumor microenvironment, like referred to by Sturgios, was a little bit surprising early on. Now I think it's well accepted. These tumors have various numbers of CD8 positive T cells. They have the chemokines that recruit those T cells in the tumor microenvironment. There's a type 1 interferon signature. People know about type 1 interferons because of alpha interferon therapy for melanoma, but local type 1 interferons are helping to prime that immune response already. And all of that is necessary to set up this endogenous immune response. So why are those tumors not rejected by the immune system? Well, we learned over the years that's because these are the tumors where the T cells are held in check by negative regulatory mechanisms such as PD-1, PD-L1 interactions, and obviously targeting that has been very successful as cancer immunotherapeutic. On the other extreme, the non-T-cell inflamed tumors lack any of these signals, and it's almost as if cancer has figured out two ways to get around the host immune system. Either let the immune cells in and shut them down, or just not let them in, because we have mechanistic data suggesting that in these cases there are defects that are leading to failed recruitment of immune cells in the tumor microenvironment. And this kind of biology can be revealed by gene expression profiling. In the very early studies, we were using Affymetrix gene chip technology. Now we have RNA-seq and the TCGA data. These are metastatic melanoma TCGA data. It's about 160 immune transcripts that characterize the T-cell infiltrated tumors. About another 1/3 are completely non-T-cell inflamed, and then there's this intermediate category that has some partial properties. And as you might imagine, having a strong endogenous immune response is a good thing if you're trying to treat with immunotherapy. These are the tumors that allow T cells to come in. Immunohistochemically, you can characterize those tumors further. You can stain for CD8 positive T cells. Even if the immune response is patchy around the periphery of the tumor, even in that local zone, it's where the CD8 T cells are located, where you can find regulatory T cells, PD-L1 expression, which in melanoma is on the myeloid cells and the tumor cells. Sergio's mentioned IDO, which is on the myeloid cells as another marker of immune tolerance. And the non-T-cell inflamed tumors lack these factors. And people used to think that the tumor set up the immune suppressive tumor microenvironment. But if there are no T cells in the tumor, you don't have these immune suppressive mechanisms. So it's actually the T cells themselves that are setting up this immune regulation as a negative feedback, and interfering with that was hypothesized, almost 20 years ago to perhaps be a strategy to restore efficacy. In our own group, it was in the early 2000s when Hanjo's lab made the PD-1 knockout mice. We thought this would be relevant for cancer. We got those PD-1 knockout mice into the lab, and showed in some of our model systems just interfering with PD-1 could be sufficient for tumor rejection. This was one of the lines of evidence arguing for PD-1 targeting. Clinically, it took a while for companies to develop antibodies and to get enthused about targeting PD-1. It's actually interesting how long it took. But now, of course, that's a big success. It's now 5 years. Maybe we should have some 5th-year anniversary, Sturgios, for anti-PD-1 being approved in melanoma, and now with approvals in something like 14 cancer entities as a single agent. As a predictive biomarker, then, this gene signature or subset of those T-cell activation genes can be a first-cut predictive biomarker for response. So, these are data generated in collaboration with Merck, and Tanguy Seiwert at our place was an active participant, now using the nanostring platform on paraffin tissue, baseline biopsies. Patients who, these are now beyond melanoma, head and neck cancer, gastric cancer, patients who lack this immune signature at baseline, they almost never respond to single agent PD-1 activity, and the responders are enriched in those that are gene signature positive. And if we're thinking about how do we make PD-1 blockade better, this allows us to conceptualize two subsets. There's the subset of patients that has a T-cell infiltrate but fails to respond to single-agent anti-PD-1. So there maybe we want combinations to lower the threshold further for T-cell activation to try and capture more of those T-cells into the reaction, right? And then you have the non-T-cell inflamed tumors, and here, if there's nothing going on spontaneously, maybe we have to figure out why that's happening, and then maybe trigger that endogenous immune response to then block checkpoints downstream from that. So that's how we've been thinking about this. And I'm just going to give you a few examples of some data, both from the lab and a little bit going forward into the clinic, how we're trying to identify new potential therapeutic targets. So one approach has been for the T-cell inflamed, if PD-1 is one of the major negative regulators, what else do those TIL express that could be contributing to immune dysregulation? So if you accept that you have these tumors where they're capable of recruiting T cells from the circulation, and then they subsequently become dysfunctional, we figured that in order to understand the biology, we actually need to profile those T cells directly in inflamed tumors. So we've done this in a mouse context and confirmed a lot of this information in human tumor-infiltrating lymphocytes. And to do this, we had some markers that we identified that were specific for tumor antigen-specific T cells in the tumor microenvironment. Many of you might not be aware that a lot of the T cells in the tumor are irrelevant for the tumor. You can find memory T cells specific for viral antigens that are not locally activated, let's say EBV and CMV reactive T cells. They're just non-specifically trafficking through. So we identified some markers that allow us to pull out the locally reactivated tumor antigen-specific T cells. There's a long history to this. It's a series of investigations. We proved in a mouse model that these are tumor antigen-specific T cells because we can transfer T cells with receptors for either a tumor antigen or an irrelevant antigen, and these markers only go up on the tumor antigen-specific T cells. So when we profile those, just to summarize here, some of the published work, those dysfunctional T cells don't just have PD-1 on their surface, and they don't just have negative regulators on their surface. We thought everything characterizing the dysfunctional T cells would be negative. But there are multiple inhibitory receptors. You know about CTLA-4 and PD-1. Sturgios mentioned TIM-3, TIGIT. 2B4 has not been targeted yet therapeutically in the clinic. This is a challenging one we're working on. But there also are a bunch, and LAG3, there also are a bunch of positive co-stimulatory receptors. It turns out that not everything that's on a dysfunctional T cell in the tumor microenvironment is negative. So GITR, CD27, ICOS, 4-1BB, OX40, almost all of these guys now are being tested in early phase clinical trials. And it's, there's a lot of work to be done still to optimize how do you drug, especially the positive co-stimulatory receptors. But when we did this, and we did gene expression profiling of those T cells, we found something like 900 new genes that had almost never been studied in T-cell biology, right? And there are some known ones that can be targeted. I mentioned LAG3. Anti-LAG3 in mice can be therapeutic, and you can combine LAG3 and other agents. This is an agonistic antibody against 4-1BB. Here we're not touching PD-1, we're not touching CTLA-4, and we have complete flatlining of these tumors. LAG3 blockade, we've been working with the BMS trial in combination with nivolumab. There's a large number of responders now in the anti-PD-1 failure cohort. There's a biomarker, the LAG3 positive T-cell tumors that's enriching for response. This is almost certainly going to be one of the strategies that moves on to become successful. But we also have these new ones. I'm just going to call this mystery molecule for now. As we identify these new candidates, we're making knockout mice of each one to see which ones have improved tumor control. This is one of these new candidate targets. We have a knockout mouse, so that factor is missing, like when we did PD-1 several years ago. And here now you put in a tumor and it's spontaneously controlled. So this is a candidate we're making antibodies against to try and move forward towards development. So that's for the hot tumor subset. So what are we going to do about the negative tumors? This is most of human cancer. When you go beyond melanoma and kidney cancer and Hodgkin's disease, the response rate to single-agent PD-1 is dropping lower, and the fraction of cold tumors goes up. So one way we can think about this is by trying to understand how is it possible that a spontaneous T-cell response is ever made against a human cancer, because there's no infection. So why are we making an immune response against a tumor when there's no pathogen in most cases, right? And then if we could mimic that, maybe we could initiate such an immune response in the cold tumors. So I briefly mentioned about the type 1 interferon gene signature that's correlating with T-cell infiltration. And just as a reminder, type 1 interferons are part of an innate immune response. Type 1 interferons are made by antigen-presenting cells like macrophages and dendritic cells when they see early pathogen engagement. And what we did then was we tried to figure out which innate immune pathway is mediating sensing of a tumor to drive type 1 interferons. And our data point to the most significant contribution of a pathway that's called the STING pathway. Anybody know about the STING pathway? This is a cytosolic DNA sensing pathway that probably evolved to detect DNA virus infection. And it turns out knockout mice that lack the STING pathway have almost a complete block in generation of a T-cell response against a tumor. So we have evidence that tumor-derived DNA ends up in the cytosol of those dendritic cells, and also that the upstream activator that's called cGAMP, cyclic GAMP, also can be generated by tumor cells and activate the pathway. So if this is how nature is making a spontaneous immune response, and this pathway is not activated in the cold tumors, what would happen if we deliberately activated this pathway with a drug? So we've done this in collaboration with a small biotech called Aduro. We had a mouse tool compound and then worked with them to identify human STING agonists, and we give a couple of injections intratumorally into mice, and these are very, very potent in these mouse models. And the mice, once they reject a tumor, if we re-challenge 2 months later, they're completely protected compared to control mice, so there's immunologic memory. This was true across 5 different transplantable tumor models, and many of you know this Aduro STING agonist and also a Merck STING agonist have gone into phase 1 testing, and now in combination with anti-PD-1. And so far we have 3 clinical responders, anti-PD-1 failures, who respond to STING agonist plus anti-PD-1. So it's not a 100% response rate. There's a lot of work to be done still to try and understand what's responsible for responders and non-responders, but this is an interesting approach that is continuing to move forward. So if STING agonists work, what about other innate immune activators? Helen Gogas knows about the oncolytic virus TVEC that's also being tested. It's FDA approved in the US for melanoma as an intratumoral injection. This also is activating innate immune pathways. And then in early phase 1/2 data, the clinical response rate is over 50% in combination with anti-PD-1 in melanoma, and this is associated with a large increase in the immune response in the tumor that's further, with T-VEC alone, that's further amplified with anti-PD-1. This was a collaborative paper between the groups working in this study, and so this and other approaches are all being tested clinically with innate immune activators, intracellular ligands for different toll receptors to initially prime an anti-tumor T-cell response, and also cytosolic RNA sensing. One caveat here is that when we're injecting these things into tumors, we really need to know if cold tumors, so the non-inflamed tumors of these different histologies, we want to be sure the receptor for these guys is actually expressed on some cell in the tumor microenvironment. So that's part of the biomarker development. So this has gone on to a phase 3 trial that's accrued, and we'll see in the coming months, year, what the outcome is of that phase 3 trial. The rest of the way we're approaching this as a discovery platform is to try to understand why there are non-inflamed tumors. And I only have time to show a few bits of data. If you zoom out and ask why might one melanoma patient or kidney cancer patient, lung cancer patient have a rip-roaring T-cell response, this is CD8 immunohistochemistry from one of my patients with a lung metastasis with melanoma. This is another patient, no T-cells in the tumor. Why? Why is one patient and his tumor full of T-cells and the other one none, right? So these tumors could be different, so we can sequence the tumors and figure out if there's a somatic difference. The hosts could be different, so there could be germline variants that are regulating the degree of immune infiltration. And then environmental differences. You already heard from Guido a little bit about the microbiota, and we have evidence that all three of these is relevant, and all knowledge about all three of these is giving potential drug targets. So I'll just show you a little bit of the data. Sergio's mentioned some of it already. We profiled melanomas from TCGA based on the presence or absence of this immune gene signature. And then by looking at the tumor exome sequencing, we found that around half of these non-T-cell inflamed melanomas have activating mutations or mutations that would activate the Wnt beta-catenin pathway. And this was published for a couple of years ago. I'm just going to summarize it in a cartoon where we built a genetically engineered mouse model that was either beta-catenin on or beta-catenin off. And this, without beta-catenin, an endogenous T-cell response was indeed generated against the tumor. And if the only difference was that beta-catenin was on, we lost that endogenous immune response, and these tumors became resistant to checkpoint blockade. And I just want to emphasize that the key to this entire puzzle is a subset of antigen-presenting cells, a dendritic cell subset that's driven by a transcription factor, BATF3. I know that's a kind of a fine detail for the non-immunologist, for the clinical oncologist, but pay attention to this dendritic cell subset because I think it's key for a lot of what we're trying to do with cancer immunotherapy. We found that tumors that lack this infiltration, that have beta-catenin on, these tumors are also resistant to adoptive T-cell transfer, so like CAR T-cell therapy. If the tumor cells have beta-catenin on, we transfer normally therapeutic numbers of activated T-cells, completely fails. Vaccination completely fails in this model. And the reason is that when beta-catenin is on, even the activated T-cells can't get into the tumor. It's like a wall. Those T-cells just can't penetrate into the tumor microenvironment. This is quantitated here, and we figured out a lot of the mechanism of that. It's because of this same dendritic cell subset being absent. So we think that the cold tumor problem that the entire cancer immunotherapy field is facing probably is not going to be overcome by new vaccine approaches or by T-cell adoptive transfer, CAR T-cell approaches. We need other strategies. So this dendritic cell subset is really important in a loop. You have dendritic cells that migrate to the lymph node that activate the T cells, but you have other dendritic cells that stay put to recruit the activated T cells back in. And so when those dendritic cells are missing, this entire cycle we think is broken. So this gives us a tool to maybe work on a new biomarker. So we're going back to human cancer specimens. So from our biobanking, And we're staining for CD8 T cells, but now in addition, we're staining for that dendritic cell subset that express this transcription factor BATF3. And I think it's not hard to imagine that there are clusters of T cells in these dendritic cells in this patient. This was a responder to anti-PD-1. This patient wasn't treated, so I don't know. That's a hypothesis that maybe this would be a non-responder. This patient's tumor has a few CD8 T cells, but no BATA3 dendritic cells. And this is a non-random distribution. If you look at spatial orientation or organization of these cells in the tumor and do kind of a mathematical model, these T cells and dendritic cells are non-randomly associating. They're deliberately getting together. And I think you need that dialogue between the T cells and the dendritic cells for the checkpoint blockade immunotherapy to work. So we're currently seeing whether this is a better predictive biomarker for efficacy of single-agent anti-PD-1. That beta-catenin pathway, we've recently published just a couple weeks ago across all the tumors in TCGA, colon cancer, bladder cancer, et cetera. We have an inverse correlation between activation of the Wnt beta-catenin pathway and immune infiltration. The mechanisms are diverse, but this argues as a refocused effort, we really need to try to work on drugging the Wnt beta-catenin pathway in novel ways to try and uncouple this problem, get T cells to go back in and expand checkpoint blockade efficacy. So we're working on that. Beyond beta-catenin, this is not the only molecular aberration in tumor cells that is linked to immune evasion and anti-PD-1 resistance. When we start marching through non-T-cell inflamed melanomas, the next most frequently mutated was the PTEN gene. PTEN is a lipid phosphatase, negative regulator of PI3 kinase, and it's a tumor suppressor gene. This was associated statistically with T-cell exclusion. And in a cohort of patients treated with anti-PD-1, anti-PD-1 efficacy was favored in tumors that retained retained PTEN. Right? So PTEN deleted tumors were more likely not to respond. And so we worked out a mouse model in collaboration with the MD Anderson group with a PI3 kinase inhibitor then, which should be, it's a pathway that should be activated when PTEN is deleted. Many of you are probably aware that PI3 kinase is also important for T cell activation. So we can't just shut down the whole pathway because we're going to lose the immune response too. So in this particular model, the beta isoform of PI3 kinase, which is not expressed in T cells, was on in the tumor. And when we drugged with a PI3 kinase beta-specific isoform of inhibitor, this synergized with immunotherapy. So this is the way I think we have to think going forward. Can we have isoform-specific PI3 kinase inhibitors to try to combine with anti-PD-1? Beyond those two, Stephanie Spranger and myself recently reviewed this growing literature of oncogenic events linked to immune evasion. There are 5 where there are clear correlates with human samples and then mouse models proving causation. All of these are hard to drug, so we put most of these in the difficult to drug category, but we have to keep working on this as a community and with the pharmaceutical industry. We have germline polymorphisms also that are associated with efficacy. It turns out some of these are also druggable. I don't have time to go through this. We have 2 germline polymorphisms that are in enzymes that can be subject to pharmacologic manipulation. So stay tuned for that. It could be that we can drug not only tumor aberrancies, but also germline to improve efficacy. So the final comment is about the microbiome. Guido talked about this already, so I'll just show a couple of slides where we knew from mouse model data and correlative human data outside of cancer that the composition of the gut microbiota impacts on immune responses at distal sites. I mean, for example, in the joint in an inflammatory arthritis, right? So we thought maybe the composition of the gut microbiota would impact on spontaneous anti-tumor immunity and therefore on anti-PD-1 efficacy. I'm just going to show one slide from the mouse data that were published a couple of years ago, where we found mice that were just raised in different facilities have different microbiotas, and anti-PD-1 either works well or minimally just depending on where you buy the mice, what facility you get them from. All of that was because of microbiota. You could do fecal transfer from the bad mice to the good mice, back and forth, and we found good microbiota could restore efficacy. We did sequencing and identified one of the bacteria types, a bifidobacterium strain, where now these are mice with unfavorable microbiota. The tumor grows quickly. If we give just the bacteria, already we start to have a therapeutic effect on par with what single-agent PD-1 blockade does, and now when we combine them together, we get complete responses. If we have mice that already have good microbiota, single-agent anti-PD-1 is enough. You don't need anything else. You get complete responses. And all of this is immune-mediated. If you get rid of T cells, you collapse all these curves. This is not the bacteria sort of deliberately or directly killing the tumor cells. It's a host immune response. And we worked out some of that mechanism where it's the dendritic cells that are pre-activated by the good microbiota. So everything is the dendritic cells in these models. So along with Laurence's and Guido's group and also Jen Wargo's group at MD Anderson, 3 groups found patterns of gut bacteria at baseline linked to outcome with anti-PD-1. Ours and Jen Wargo's were done in melanoma, and there were bacteria sequences enriched in responders and other bacteria sequences enriched in non-responders. As a predictive biomarker, you've probably heard about mutation load. We talked about immune gene signature, quantitative immunohistochemistry for CD8 T cells. We wondered if just the gut microbiome sequencing could be a predictive biomarker. And in this cohort, we only had 15 patients at this time where we had both tumor and gut microbe sequencing. But the best predictive biomarker was the microbiome. So we had quantitative PCR of different species. If patients were positive, you could almost completely separate with 2 different assays, responder from non-responder, which is more an extreme separation than mutation burden or PD-L1 or CD8 immunohistochemistry. So it's a small cohort and it's very early and everything needs to be confirmed, but it really argues that the composition of the gut microbiota can impact on the overall sort of immune primability in the patient and anti-PD-1 efficacy. And like Guido mentioned in their model, this can be transferred into mice. So if you take the same tumor, genetically identical mice that are germ-free, the same PD-1 antibody, but the only difference is whether the microbiota came from a responding patient or a non-responding patient. If it came from a non-responding patient, anti-PD-1 doesn't work in the mice. If it came from a responding patient, the immune response is a little stronger already, but now anti-PD-1, PD-L1 works. So this argues there's a causal role for those bacteria. And this has led to identification of one of the first human bacterial entities. This is the last end of the discussion here. Where now we're trying to give that bacteria back. That's a biotech company in Boston called Evelo. They've generated a clinical grade bifidobacterium now to try to give back. We're doing this with anti-PD-1 naive, but also a cohort of anti-PD-1 refractory patients. We know more than 85% of metastatic melanoma patients have no detectable bifidobacterium. So we're just giving it back is going to be interesting to see if it takes, can we modulate the immune response, and see if by chance just the single bacterium can restore anti-PD-1 efficacy, although that is a work in progress. And I think we're going to learn a lot from these and other similar studies. Some groups are doing fecal transfer also from responders into non-responders to see if that improves efficacy. So I think we're out of time. I'm going to show you 2 slides, which is we're starting to think about secondary resistance. So patients respond, their tumor shrinks, they do well for a period of time, and then they have a new tumor appear, a tumor that grows. Many of those patients have antigen loss or class 1 MHC loss, so they become sort of invisible to the immune response. Some of those tumors, though, become T-cell negative. They start off T-cell inflamed, and then they become T-cell non-inflamed. So we've been profiling some of those. We have the first 2 patients. The first of these was CD8 infiltrated. The recurrence is CD8 not infiltrated, and they lack the gene signature. This was a beta-catenin aberration. So started off beta-catenin negative, now rip-roaring beta-catenin positive. So secondary resistance apparently correlating with beta-catenin pathway activation. The second one, this was a patient treated with ipi-nivo, responded, recurred, and now the new sequencing of the new tumor The only new aberration is PTEN deletion. So these two pathways that we had identified already, we seem to have immune selective pressure to drive outgrowth of tumors that have a new molecular aberration. So this is by immunohistochemistry. This tumor started off CD8 infiltrated and PTEN positive. Now it's PTEN negative and it lacks CD8 cells. So again, arguing maybe if we can drug these pathways early, maybe we can also prevent secondary resistance. So I forgot I threw that in there at the end. So sorry about the false advertising that it was the last data slide. So I've tried to argue that just studying the biology of the T-cell inflamed tumor microenvironment has been very useful for us. By studying those dysfunctional TIL, we have additional new targets, some of which are in the clinic, some of which are very early, and we'll see will go to the clinic. Innate immune activators in the tumor might be one way to turn cold tumors hot, if you will. This is ongoing. Molecular aberrations in the tumor are associated with either primary or secondary resistance. We need to find new ways to drug these pathways, and then by putting together these different bioinformatic sequencing data, so multidimensional omics from patients, we're learning about new strategies, including the microbiota that might be modifiable therapeutically. I'll leave the acknowledgements here. I think I had the names on each of the slides of people in the group and in the clinic. And I wanted to remind people of our band. We play in a band. I play guitar. It's called the Checkpoints. And Rachel Humphrey, who was the medical lead who took ipilimumab to the clinic, is our singer, right? Patrick Hu, who's the chair of oncology at MD Anderson, plays keyboards, right? I play guitar. And our harmonica player is Jim Allison. And then Rachel herself wrote this little piece, what to do when your harmonica player wins the Nobel Prize. We thought this was the only time that this had ever happened. It's probably true it's the only time in medicine, but then we realized that Bob Dylan has a Nobel Prize and he plays harmonica, but that was for literature, right? Not for science. And anyhow, so we play at the ASCO meeting every year. This was last year at Buddy Guy's Legends, and it'll be, if you do go to ASCO, any of you, this year, Sunday night of ASCO, we're playing again at Buddy Guy's Legends. Thanks.
Thank you, Tom, for this wonderful talk. And I would say I'm really honored. I heard your paper before it was published in Science in the IMWG. I do remember that moment about all the stool transfer. And you must be very proud because, in Israel, Jacob Shatter with Gal Marker, they are performing this study from patients that are extreme responders in immunotherapy. They are in patients that have failed immunotherapy are doing transplant.
Fecal transplant.
Yes, yeah. Yes. So that's really wonderful. Any questions?
Any burning questions? The repertoire of the band, what's your favorite song? You can make requests at ASCO, right?
He makes capsules.
The capsules, yes. So this actually was an interesting process. So it hadn't been done before, right? At first, the way some of these studies are done are thinking of the bacteria as a probiotic, which is like a food supplement. It's not a drug, right? But if you're really using a food supplement that's not quality controlled, you can't claim efficacy in a cancer context. So it had to go to the FDA. To go to the FDA, it has to be a sequence-verified, stable bacteria, and it's packaged in a capsule, monoclonal bacteria, right, that survives the gastric pH and then opens in the small bowel. It had to survive a stability test, so it had to be put on the shelf for 6 months, and then you resequence and retest, make sure it still has potency. It needs a potency assay, so the potency assay is human dendritic cells activated with it to show that they make IL-12, and all of these things, right? You have to have zero contamination, you have to have zero pathogens, all of these things controlled. So it took a while for, Evelo, the biotech, did all of this, right? So it's a long way from like a simple laboratory experiments to the clinic. I should add that, there are early data that over-the-counter probiotics are not sufficient to have this effect. And the early data are suggesting that patients taking probiotics are probably less likely to respond to anti-PD-1. So not just any bacteria. I'm not sure if Greek yogurt, like Faye, this is probably okay, right? I had some this morning. Yogurt is it's a food. I mean, it's got to be good for you. But I feel better when I have yogurt, right? These over-the-counter probiotics that have never been tested for anything, but people are selling as maybe a cancer treatment, completely unproven. We have to be super careful. And in my clinic, I advise patients not to take any probiotics, but if they want to do something, to have a balanced diet that includes a lot of fiber with fresh fruits and vegetables. Like, I'm sure a Greek salad, things like this would be great recommendation. So Faye and a Greek salad, that's me. All right. Thanks.
Oh, I get a diploma.
So this is on behalf of the Institute of Molecular Medicine and Biomedical Research. You got this Honorary Black to Professor Thomas Gajewski as recognition of his long-lasting offer in scientific research.
Okay. Thanks. I wanted to give you access to the microphone. So it's beautiful. Yeah.
Thank you very much for supporting the Congress.
Yes, thanks for inviting me. Yeah.